Hi Year 12s. In this video we're going to unpack the role of CRISPR-Cas9 in bacteria cells. So this comes from the key knowledge point that the function of CRISPR-Cas9 in bacteria and the application of this function in editing an organism's genome. So we're going to split this into two videos. The highlighted part there, just the function of CRISPR-Cas9 in bacteria is what we're going to focus on in this video. And then in the following video, you'll look at how we can manipulate that to edit another organism's genome, which is pretty cool. So you will know if you're successful in this lesson if you can outline what happens when a bacteriophage enters a bacteria cell. You'll also be able to describe how the Cas9 protein uses complementary RNA sequences to locate and cut specific DNA sequences. You'll also be able to explain what a CRISPR sequence is and how that helps bacteria cells to defend against future bacteriophage infections. So let's start with that bacteria and bacteriophage relationship. So obviously a bacterial cell is a quite simple cell. We know that the DNA is found in the cytoplasm. It says here nucleoid, that's a region of the cytoplasm that DNA is found in, and they have a pretty simple chromosome which is circular. A bacteriophage, you can see here is this green thing in that diagram on the right hand side. So it's not a diagram, it's more of a visual representation of what we think these things look like based on the evidence that we have on electron micrograph images. So it's basically a virus that exclusively infects bacterial cells. So the way it works is it will attach to that bacterial cell, it'll inject its DNA, hijack that bacteria's, protein synthesis strategies to make more virus particles, kill that cell. Those virus particles are then able to go and infect other cells. So the way that bacterial cells have, adapted their own little immune system against these bacteria cells is using this Cas9 protein. So we're breaking up that CRISPR-Cas9, looking just at Cas9, and we'll look at CRISPR in the next slide. So Cas9 is an enzyme. It's an RNA-guided enzyme, and it's a DNA endonuclease. So let's unpack those two things specifically. The fact that it's RNA guided means that it uses an RNA sequence to find a specific sequence of DNA using base pairing rules. So you can see here in this image on the right that this little yellow part of this protein is RNA. That bacteria Cas9 enzyme is using to find a specific sequence of DNA. It's unraveling the DNA, which is that blue part, to find the specific sequence that it's looking for. That yellow part, that's part of that protein. It's part of the Cas9 enzyme. The fact that it's an endonuclease means that it cleaves and cuts DNA. So what it's doing is it's searching using the complementary base pairs of the RNA molecule that's embedded within it to find a specific sequence of DNA to be able to cut that. So it's trying to find the bacteria DNA within its cell. When it finds the bacteria DNA, it's chopping it up and trying to kill it so that it can't infect and hijack its machinery to make more virus particles. So the bacteria cell is actually able to program that enzyme to cut any sequence it wants, and we'll go into that, how it does that in a moment. But basically what it does is it unzips DNA and moves along it to find that specific sequence. And you can see that in this GIF here. The red part there is the RNA that's embedded within the protein, and the blue part is the DNA, and it's found that specific sequence, it's cutting it up, and that DNA is now being removed from that enzyme because it's already done its job. The RNA stays embedded as part of that protein. So it's RNA guided because it's got that RNA within the protein. It's helping find a specific sequence of DNA, and it's basically just cutting it up to make it unviable. CRISPR, this is the part that helps, I guess, the bacteria cell find that specific sequence. So a CRISPR is a sequence of DNA within the genome of the bacteria cell itself. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So the clustered part means that there's groups of them. Regularly interspaced means that there's something in between them. And short palindromic repeats means that it's the same backwards and forwards. And obviously repeat means that it happens over and over again. Okay, so let's now look at how CRISPR works, or what CRISPR actually is. This here is a representation of a bacterial cell. So the yellow outline there is that bacterial cell, and the black stuff in the middle, that is the bacterial chromosome. So we know that it's a circular chromosome. It's not exactly circular, but we know that it has a closed loop. So it has these CRISPR regions within that genome of the bacteria. So the CRISPR regions, I guess, are those palindromic sequences that are punctuated by what we call spacer regions. Basically, what's happened is these bacteriophage, when they infect a cell, they inject their DNA, and that's how they try and manipulate the bacterial cell to translate proteins that make more viruses. What the bacteria is doing every time it gets infected by one of these bacteriophages, it's keeping a little bit of its DNA and it's inserting it in between those CRISPR regions. So what it can then do when they have future infections is use that piece of DNA, or that CRISPR DNA, to create CRISPR RNA or guide RNAs. That word, or those abbreviations, can sometimes be used interchangeably. Basically, how I remember it is that CRISPR regions, or CRISPR RNA, is when it's sort of sitting like this, it's been transcribed into this RNA. Once it's been absorbed into or incorporated into the CAS9 protein, we then call it guide RNA. So once it's part of that CAS9 protein. So again, that is something that should be added to your glossary, along with all the other new words like CRISPR, Cas9, all those things that we're learning today. So it translates that little bit of DNA, makes it into CRISPR RNA. Once it then gets embedded into the Cas9 protein, we now call it guide RNA, or gRNA. It's then able to locate, cut, and cut complementary target DNA sequences. So you can see down here on the right, this Cas9 protein has been able to use that guide RNA to find a little bit of bacteriophage DNA and chop it up before it's able to fully hijack that cell. Again, you can see that in this picture here. Can I rub out my stuff? Yes. That this green stuff here, so this is a Cas9 protein. The green stuff, that is the guide RNA. And you can see that it's coming along. It's unraveling that DNA. Remember that DNA has hydrogen bonds between the base pairs. Super strong, holds them together, but it's also able to be unraveled quite easily. So the Cas9 protein is taking advantage of that, unraveling the DNA. Once it finds a sequence that's complementary to its RNA, it says no, and it chops it up so that that DNA is no longer viable to infect the cell.